You may begin, Dr. Harris.
Welcome to the third session of the 20th year of the NIH course entitled Demystifying Medicine. The logo, as shown on the slide, is the Brooklyn Bridge. We are like the individuals on the bridge who are discussing or learning from one another about important biomedical and engineering progress as it can influence medicine. Things are never the same after a bridge has been successfully built. And this course, in effect, may be thought of as a course in bridge building. The next slide provides some details that you may require. The goal is to bridge the exciting developments in biology and engineering with medicine. All sessions will be virtual and are held on Tuesdays from 4 to 6 p.m. Eastern Standard Time from January 4th to May 3rd. The location for the video cast is indicated on the slide, and you can see a copy of the course schedule by Googling Demystifying Medicine or going to our website, which is indicated. Those who attend at least 50%, which is 10 sessions, and pass a computerized final exam can receive a certificate. For physicians who seek CME credit, today's course credit number is listed 38643. We urge you to, would you go back, please? We urge you to submit questions during the two presentations, and that can be done via the send live feedback on the video cast display on your computer. Please know that all previous sessions from the very beginning are archived on the NIH Videocast Network, past events. Next slide, please. Today's topic is the intestinal microbiome. I think that Leeuwenhoek in the 17th century was the first to ever see the microbiome by examining contents of fecal material on his miniature microscope. In the 19th century, Metchnikoff is probably the first to relate it to disease. In that sense, what today we would call toxic shock. But he also related it to the possibility of health, and that by modifying the microbiome, there may be influences on human health. But it wasn't until 1977 when Carl Woese, who discovered archaea, introduced 16S ribosomal RNA sequencing, which revolutionized microbiology and permitted phylogenetic taxonomy to replace descriptive cultural activity, ability to grow and culture of different microorganisms. Now, the discovery of these methods led to the quantification of microbiome species, and then the study of its composition, regulation, and role in physiology and disease. However, for much of the work, we are in a descriptive stage. That is, describing the microbiome in different ages, socioeconomic groups, and comparisons in control patients and those with various diets, weight, and disease. The huge challenge is to determine whether the observed microbiome differences represent cause or effect. Now, the availability of germ-free and gnotobiotic animals has aided. However, the ultimate questions concerning humans, in whom clinical studies of cause, effect, and mechanism are difficult, if not at times seemingly impossible to perform. And this is the bridge that we will be discussing today. Today, we're very fortunate to have two outstanding scientists discuss their exciting work in bridging the microbiome with two major clinical problems: malnutrition and cancer. Now, our first speaker will be Jeffrey Greten, who graduated from the University of Chicago School of Medicine, trained in medicine at Washington University and St. Louis, and in gastroenterology, and in biochemistry at the National Institutes of Health, and then returned to Washington University, where he has spent his entire career. Jeff started to use genomic sequencing, mutagenesis, gnotobiotic animals, and their response to diet and functional assays in preclinical and clinical studies. In one of his papers, he says, "I want to confess I am more microbial than I am human, because the contents of the microbiome, numbering in the trillions, far exceeds the sum total of all of the cells in the human body." Jeff is a distinguished university professor, the Robert Glaser Chair Distinguished Professor at Washington University, and professor of pathology and immunology, also of developmental biology and of molecular microbiology. He has received an extraordinary number of the highest honors that can be bestowed upon a scientist. He's a member of the Association of American Physicians, a member of the National Academy of Sciences, the American Academy of Arts and Sciences, the National Institute of Science, the Institute of Medicine, the American Philosophical Society. And he has been recipient of virtually every major prize and award given for biomedical research. One of those awards in 2018 was from the Royal Society, which bestowed its Copley Medal, which is the oldest scientific prize in the world. And I would just read, as put on the slide, the reasons for that award. It's because his work led to a fundamental shift in the way scientists understand the relationship between microbes, health, and disease. Greten, who graduated in medicine in Germany, was a postdoctoral fellow at Johns Hopkins in tumor immunology, which is his main interest, returned to Germany, and then in 2010 was recruited to the National Cancer Institute, where at the present he is the deputy branch chief for the Thoracic and GI malignancy branch. He's a tenured senior investigator and co-chair of the Center for Excellence in Immunology at the NIH, and also of the large program, interdisciplinary at NIH, directed toward hepatocellular cancer. His major research has involved tumor immunology, focusing on hepatocellular cancer. And during this work over the past several years, he's become increasingly involved in the relationship between liver tumors, particularly cancers, and the gut microbiome. And the topic of Tim's talk is how the gut microbiome controls growth of liver tumors. Now, we will consider questions and answers and discussion at the conclusion of Dr. Greten's talk, but please submit your questions during the entire program, and they will be presented at the end. So now, I want to thank both of our speakers for taking the time and interest to participate in this program. And Jeff, if you would please begin.
Well, thank you very much, Wynn. It's a privilege and pleasure to be able to participate in this wonderful program. I hope everybody can see my screen. This is
no,
a story.
Not yet, Dr. Gordon. You're sharing?
Try one more time.
Okay.
Has sharing been achieved in this world? How about now?
Yes.
This is an adventure. Two groups of individuals. On the left, you can see a group of very talented students and postdoctoral fellows in the lab, which is located at the Center for Genome Science and Systems Biology at Washington University in St. Louis. And our longstanding collaborators led by Tamid Ahmed at the International Center for Diarrheal Disease Research. This is work funded by the Bill and Melinda Gates Foundation and the NIH. I have no conflicts to report. It's work directed at the development of complementary foods for treating childhood undernutrition. Now, this is a pressing, vexing, and deeply troubling global health challenge made worse by the current pandemic. It's the leading cause of death of children under the age of 5. There have been many, many epidemiological studies that have demonstrated that childhood undernutrition is not due to food insecurity alone, but represents the confluence of many factors that operate within and across generations. Current therapies have evolved to reduce mortality, but they've had very limited, unfortunately, success in overcoming the long-term consequences or sequelae of undernutrition, which includes impaired linear growth or stunting, defects in immune responses, including to vaccination, and neurodevelopmental abnormalities that impair cognition. So there's something about pathogenesis that we're missing. And what is that? Well, we're also missing a comprehensive definition of the biological state or states of undernutrition. The typical way of defining undernutrition is through the use of anthropometry, the measurement of height or length and weight at a particular age, and the referencing of those values for an individual to a multinational birth cohort assembled by the World Health Organization. And the definition of deviance from normal based on the number of standard deviation an individual is from chronologically age-matched healthy individuals in this multinational birth cohort. So we can define stunting as a length for age score that is a certain number of standard deviations. Moderate stunting, 2 to 3 standard deviations below the median for this reference healthy cohort. Wasting or impaired ponderal growth as a deviation from normal of weight for length or height, a WLZ score. Moderate wasting, 2 to 3 standard deviations below. Severe wasting, a WLZ score that's more than 3 standard deviations away from normal. But of course, who would define diseases like cancer, or cardiovascular impairment as moderate or severe. We need a much more comprehensive definition of this state of impaired growth. And you'll see why this is particularly important when it comes to connecting the microbiome to disease pathogenesis. So what are the hypotheses that our group at Washington University and the ICDDRB, the International Center for Diarrheal Disease Research, are pursuing? Well, the hypothesis is that there is a definable normal, that the gut microbiota has a characteristic program of assembly or maturation that begins at birth. And that disruption of this developmental process affecting an organ, a microbial organ, is causally related to undernutrition. Now, a corollary is that healthy growth of infants and children is linked to healthy development of their gut microbiota. That there has to be a synchronized co-development of this microbial community and the host. So I'm going to take you on a multi-step journey that Dr. Arias has alluded to in his introductory comments. A journey, I think, that is emblematic of the journey that many people in this field are pursuing. The first is to define normal in a given population and ask whether that definition of normal generalizes to other populations. The second step is to develop a metric or metrics for quantifying deviations from normal and determining whether those deviations correlate in a statistically significant fashion with disease and disease severity. The third step, that Dr. Arias emphasized, is to determine whether, if deviations from normal do correlate with disease, whether those deviations are simply an effect or perhaps are a cause of disease. The next step is identifying therapeutic targets within a microbial community, building confidence and rationale for that selection, and then developing therapeutic interventions that are not only efficacious in a given population, but that are safe, durable, and generalizable, and culturally acceptable and affordable. So I'm going to take you on this journey that we have been pursuing, and we'll begin in an urban slum, Mirpur, located in the capital of Bangladesh, Dhaka. You can see from this particular slide that the population density in this urban slum is remarkable. 500,000 people living within 2.5 square mile area. Population density of 77,000 per square kilometer. And the definition of normal was pursued in a birth cohort where individuals were enrolled at birth, reflecting the deep trust and commitment of mothers and families for the health of their offspring, as well as the deep trust and respect that they had for the healthcare providers that they have been interacting with, so that longitudinal studies could be performed successfully. Tamid Ahmed and his colleagues have forged that level of trust, respect, and devotion over the course of the last 4 decades. Members of this birth cohort were followed monthly, initially for the first 24 months of postnatal life, and ultimately through the first 60 months of postnatal life, with monthly sampling of the fecal microbiota. In addition, monthly anthropometric data were accumulated, and the initial focus was on those infants and children that had consistently normal anthropometry during the first 2 years of postnatal life, a subset of members of the birth cohort. And then the application of culture-independent methods that Dr. Arias had described, using the bacterial 16S ribosomal RNA gene as a barcode of life, of bacterial life, and classifying organisms based on the 16S sequencing results. Now, from healthy members of this birth cohort living in this urban slum, Mirpur, a microbial picture of normal community development emerged. And in one iteration of this picture, shown here, you can see the columns as age bins, the rows at different bacterial taxa, and the colors indicating the relative abundances of these organisms as a function of time after birth. And you can see down in the left lower quadrant of this heat map, a group of organisms that are very prominently represented in the first several months of postnatal life and become more diminutive, and those that become more prominent later on. Now, this is the result actually of the 5-year analysis. This gives you an idea of what parts are there and what parts might be shared by members of the microbial community. But they fail to really address a fundamental aspect that we need to understand about the gut microbiota. And that has to do with its organization and its dynamism. So we need to go beyond a simple list of parts to identify interactions between components. Now, the number of potential interactions that might occur in such a community, even if you had 100 bacterial members, is literally astronomical if you consider all possible pairwise or higher order interactions. So we need to identify through feature reduction a smaller number of interacting components that describe a normal microbial community development in healthy individuals. Now you might ask, why, Jeff, are you describing this need for looking at interactions? So I'll try to paint a picture of why by going to macro ecosystems and even human behaviors. So imagine if you only focused on individual fish and discarded any consideration of their interactions, you would never appreciate the concept of fish schooling. Similarly, you would never be able to appreciate the dynamism and the organizational properties of birds in flights or ants foraging. So how are we going to be able to do feature selection and identify a smaller number of parts, interacting parts in the gut microbiota that might describe across a population normal community assembly? For that purpose, we actually turned to the stock market and a field known as econophysics, where individuals started to use statistical methods to look at covariating price of stocks to understand the interactions between the activities of different economic sectors and the behavior of the stock market. And a very talented postdoc in the lab, Arjun Raman, applied the principles and methods of econophysics to identify a small group of 15 covarying gut bacterial strains that described in this healthy cohort of infants and children living in the Mirpur urban slum normal community development. And this network of covarying taxa are described here in this, diagram here. And the green members are components of this network, which Arjun termed an eco group. From this analysis, as well as from other types of analysis, we were able to determine this program of community assembly in healthy individual is largely complete by the end of the second postnatal year. We also discovered, Arjun found in his initial analysis, that these taxa are also represented in the developing gut microbial communities of healthy members of birth cohorts living in other low and middle income countries. So there's something about a shared feature of this definition of normal that could be captured with this approach. Using this eco group, we were able to discern that children with undernutrition had gut microbial communities that looked younger than you would expect based on their chronological age. this can be viewed as a microbiota immaturity. And the degree of immaturity was greater in children with moderate acute malnutrition, those having WLZ scores between -2 and -3, than in children with severe acute malnutrition, those having WLZ scores that were more than 3 standard deviations from the median of the WHO reference birth cohort. And critically, this immaturity was not repaired with current therapeutic foods. That was the result of several human studies that were done with our colleagues at ICDDRB. But perhaps this is not surprising because current food-based interventions for childhood undernutrition have not been developed with knowledge of or consideration of the developmental biology of the microbiota. So these children were left with a persistent developmental abnormality affecting one of their organs, microbial organ. We wondered whether there is something that we could do about this. But before we proceeded, we listened to the words of Dr. Arias and tried to capture this concept of, can we perform a test that distinguishes between effect or cause? So Laura Blanton, who's a very talented graduate student in the lab at the time, together with Maria Carlson and Dave O'Donnell, who established our Notobiotic facility here at Washington University in 1996, did a series of microbiota transplant experiments. So a group of chronologically age-matched healthy individuals and those that had undernutrition, as defined by anthropometry, provided fecal samples. Those samples were preserved immediately after they were provided, and they represented a snapshot of the microbial communities of the donor. Each fecal sample from these chronologically age-matched individuals were transplanted or introduced into the intestines of young germ-free animals. So basically, several animals received the microbiota of a given individual, and several microbiota were tested in this fashion from healthy versus undernourished donors. And the test really consists of taking this snapshot and replicating the snapshot of microbial life in multiple animals, and then converting the snapshots to a movie so that we could follow over time after transplantation the effects of those communities on the biological properties, physiology, metabolism, immune function of the recipient animals. The animals were given diets that represented the diets of the human donors. So the diet that was typical of children living in Mirpur were given to the recipients of both the healthy donor microbiota as well as the undernourished donor microbiota, so that we could reenact the intersection between diet and microbial communities, and ask the question, how many of the phenotypes of these undernourished children could be transmitted to recipient animals? And the answer was many. So using quantitative magnetic resonance to define body composition, using microcomputed tomography to quantify bone growth, using targeted mass spectrometry to characterize metabolism, and using a variety of approaches, including flow, to characterize immune function. It was determined by Laura in this first set of experiments and subsequently by others in the laboratory that many features of undernutrition could be transmitted to these recipient animals. Now, what Laura did was to take all recipients, all mouse recipients of all donor microbiota in the first series of experiments that she performed, and apply machine learning algorithms, random forests, to identify organisms whose representation was significantly associated with different facets of growth. So she identified a subset of growth discriminatory strains from these experiments. And these strains included strains that were underrepresented and presumably underperforming in immature microbiota. And they became our therapeutic targets. Now, we're funded by the Bill and Melinda Gates Foundation. We wanted to develop microbiota-directed therapeutic candidates using these animal models that portrayed the microbes of the very population we want to treat, as well as their diets. But we had to develop an intervention that was culturally acceptable, affordable, and scalable. So we thought, we shall not start by basically creating a next generation consortium of anaerobic cultured bacteria, which would be difficult to preserve in a cold chain. And there's not a lot of technology about how to do so. And this would be extremely expensive, as well as facing a variety of regulatory hurdles that wouldn't be faced if we were to turn to food as a principal and first choice for repairing the gut microbial communities of these undernourished children. So, a second series of hypotheses. Perhaps the foods that are consumed between the period when exclusive milk feeding ceases and a child is fully weaned, the period of complementary feeding, some of these foods may contain key nutrients that are coveted by these underrepresented and underperforming growth promoting organisms. Now, these foods by definition are available and culturally acceptable because they are the foods that are given to children in this area of the world. But these types of foods also embody the choice that every family around the planet has to make. What food should I select to ensure the healthy growth of my infant or child? So in approaching this mining expedition of identifying complementary foods, we're really approaching this set of decisions about how to feed our infants and children. Decisions that currently are based on policies that are not predicated on knowledge or consideration of the developmental biology of the microbiota. So the approach that Jeanette Gehrig, who is a very talented PhD student in the lab, in concert with a talented postdoc, Sid Venkatesh, a talented PhD student, Hao-Wei Chang, Mike Barrett, person who oversaw our interactions between the clinical studies being done in Bangladesh and our clinical work here in the labs at Washington University, and Tamid Ahmed conducted a screen for these complementary food ingredients that might promote the fitness and express beneficial functions of our therapeutic targets, and to incorporate these ingredients into what we would call microbiota-directed complementary food prototypes that we would test first in our preclinical models. So here is our strategy for testing. We cultured organisms from the fecal microbiota of the children representing our target population. And these were age and growth discriminatory organisms. And the consortia of cultured sequence organisms were introduced into germ-free mice. And these germ-free mice were fed complementary foods available to children living in Mirpur. And these foods were tested individually and in various combinations to identify those that would increase the fitness initially of our target growth promoting organisms. If we got a hit, we would prioritize the hits based on the sustainability, the affordability, as well as the organoleptic properties of the foods, their taste, texture, and smell. Because if they didn't have satisfactory organoleptic properties, they would never be accepted by the mothers and, most importantly, by the children that are the targets for all of this effort. Now, after this screen was initiated, sequenced bacterial strains from children living in Mirpur, but rather the intact uncultured microbiota of children who had been treated with standard therapy, but whose microbiota hadn't been repaired. So could these MDCS, microbiota-directed complementary foods, repair the microbiota that had been introduced into these animals? And for those combinations that did, additional mechanistic studies were performed about how the community responded and how the host responded. And a decision was made that I think embodies some of the challenges in the field. Do you directly go from mice that, even though they portray the microbes and diets of the target population, don't have physiologic and metabolic properties that are as close to humans as certain other animals, and you choose a second animal species? And that's something we did. We developed methods for rederiving pigs at birth as germ-free and maintaining them in a germ-free state, and introduced these consortia of organisms from the children of Mirpur and looked at the effects of our most leading MDCF candidates. So those candidates were then advanced to our first pre-proof of concept study. It involved children aged 12 to 18 months with WLZ scores of -2 to -3, which you'll now know is the score assigned to children who have moderate acute malnutrition or MAM. These children in this small study of 15 per arm received one of 4 different foods, food supplements, a ready-to-use supplemental food, rice and lentil based, that's a commonly used nutritional intervention. And then we had the various MDCS, MDCF 1, 2, and 3. Children were enrolled. There was a run-in phase for 2 weeks in which anthropometry data were collected, fecal samples were obtained, and a blood draw performed just at the end of the second week. This blood draw was very important because we wanted to get a more comprehensive definition of the biological state of these children prior to and at the end of the intervention. And we could do so because there's an aptamer-based technology developed by SomaLogic, which allowed more than 1,300 different proteins, which are both biomarkers and mediators of many aspects of host physiology, to be quantified. So the intervention, excuse me, we're almost there. We're going back in time. The intervention consists of 2 feedings each day under supervision in a nutritional rehabilitation unit established by our colleagues at ICTDRB right in the middle of the urban slum. And the devotion of the mothers to children is embodied in one measure by the fact that they brought the children twice a day for these interventions, which were performed under supervision. There was a 2-week follow-on period of observation after the intervention. And the outcome measures were, what are the effects on the gut microbiota and what are the effects on the plasma proteome? We did not expect, given the short duration, 1 month, and small number of children, for us to achieve a statistically significant effect on either ponderal growth or linear growth. So these are the results of this pre-POC study. You can look to the lower left and see in a PCA plot the different configurations of the microbial communities of different groups of children. In this lower portion of the PCA plot are children with severe acute malnutrition who are untreated. They're 12 to 18 months of age. Here we see far away the configuration of 12 to 18-month-old healthy children living in Mirpur. These configurations are defined by the representation of these 15 eco-group taxa that had been identified as consistently co-varying and as a way of defining the developmental biology of the microbiota. You can see that children with moderate acute malnutrition or untreated are closer to healthy than those who had SAM and were untreated. But you can see of all of the intervention, MDCF1, RUSF, or MDCF3, that after 1 month, their effects on the configuration were significantly less impressive than the effects of MDCF2, which, by the way, was the best performing MDCF in our notobatic mouse and piglet models. not only was there repair of the microbiota, but also a dramatic effect on the plasma proteome. And on the right side of this slide, you see a group of 50 proteins that were identified from this aptamer-based analysis of the plasma proteome of healthy children and untreated children with SAM as most discriminatory for a healthy state. And the colors indicate the change in the levels of these proteins as a function of different treatments. So the levels, the treatment effects is row normalized, and movement towards red indicates movement towards a more healthy level of this protein. MDCF2, assuming you're not red-green colorblind, is associated with this dramatic change in the level of these healthy discriminatory proteins that represent many different aspects of human physiology. They are biomarkers and mediators of bone growth, of metabolism, of immune function, and of neurodevelopment. So with this pre-POC study in hand, a second proof of concept study was performed with MDCF2, our lead, and RUSF, the commonly used intervention. You can see the complementary foods that are represented in MDCF2: chickpea flour, peanut flour, soybean flour, green banana. In RUSF, it's principally rice and lentil and some skim milk. There's no skim milk in MDCF2. It's very costly. And you can also see that there is micronutrients in both equivalent amounts, that the protein energy and fat energy ratio and amount of carbohydrates are roughly equivalent, but that the caloric density of MDCF2 is 15% less than that of RUSF. So as in the pre-POC study, a POC study was performed where under supervision, 2 servings, 25 grams each, was given per day of either MDCF2 or RUSF. Now here's the design of the study, a more highly powered study, a longer study, 3 months. Initially, twice a day administration of either MDCF2 or RUSF in a clinic under supervision. The next month, once a day in the clinic, once a day at home under supervision. And the third month, twice a day at home under supervision. Again, 12 to 18-month-old children with MAM, moderate acute malnutrition. There's a 1-month follow-up. The primary outcome measure is a change in WLZ, and the secondary outcome measures alterations or repair of the microbiota and alterations in the plasma proteome. This study was led by Tamid Ahmed and Ishita Mustafa, and I'll present the results in the next slide. Robert Chen is a very talented MD PhD student in the lab. He analyzed the anthropometric results using linear mixed effects models that controlled for gender, previous illness, and a random effect for each patient. And you can see that MDCF2 produced a significantly greater rate in change in WLZ score, a greater rate of ponderal growth compared to RUSF, even though MDCF2 is 15% less calorically dense. So we now should think about therapeutic foods not just based on their caloric content, and we should ask, what are the microbial and molecular mediators of the superior growth effects of MDCF2? For that, we did an analysis. Robert Chen did an analysis where he took the proteomic data set from all participants in both arms and then correlated changes in WLZ with the change in each of 5,000 different plasma proteins whose levels were measured prior to and at the conclusion of the intervention. So shown here in the lower left is an example of the types of tests of correlation between the rate of change in WLZ over time and the rate of change of a particular protein, IGF-1. And those plots for each individual are shown as dots. And from this analysis, a group of 70 WLZ correlated proteins were identified. They're involved in many aspects of postnatal growth, musculoskeletal development, immune function, CNS development. And in this plot developed by Robert, we're looking at, on the y-axis, the fold change of each of these 70 proteins produced by MDCF2 compared to RUSF. And the vast majority of these proteins increased more with MDCF2 than with RUSF. So now we have a group of 70 proteins that are impacted more by MDCF2 than RUSF that we now know are instrumental in various aspects of postnatal growth. Similarly, Matt Hibbert looked to the microbiota to identify WLZ-associated bacterial taxa and to determine whether their representation was more impacted by MDCF2 compared to RUSF. Again, for each individual, taking the data about the change in WLZ over time and correlate with the change in abundance of several hundred different bacterial taxa identified by 16S sequencing using linear mixed effects models. And those results shown here disclosed 23 different taxa that were defined as statistically significant in terms of their associations with WLZ. And 21 of them are positively correlated, 2 are negatively correlated. This analysis, shown in the lower portion, lower half of the slide, describes a coefficient, beta 1, from these linear mixed effects models that represents the change in WLZ for a unit change in the abundance of a given taxa. The taxa are described by amplified sequence variants. These are determined from 16S sequencing. And you can see Faecalibacterium prausnitzii has the greatest impact on WLZ for a unit change in its abundance, but there are a whole host of these taxa, including Prevotella taxa, etc. But going back to the original question, MDCF2 has a significantly greater effect, produces significantly increased amounts of these positively correlated taxa than does RUSF, and has the greatest effect on reducing the abundance of the principal negatively correlated taxa, Bifidobacterium longum. So this type of analysis, it starts to connect the dots. How are changes in ponderal growth and an MDCF2 intervention related to changes in effectors of different aspects of growth, as well as changes in the representation of different components of the microbiota. Now, the next task, of course, is to get deeper into the mysteries of the microbiota and to understand how these different taxa change their patterns of gene expression. So how do expressed genomic features of WLZ-associated strains correlate with growth responses? That question and the ensuing answers are absolutely critical for more detailed mechanistic insights about how MDCF2 and other food-based interventions would be affecting the microbiota and host growth, key for developing improved MDCF, as well as for the discovery and use of symbiotics, which are combinations of pre and probiotics or probiotic species. So the tasks here are to reconstruct the genomes of WLZ-associated strains present in the fecal samples from children in both arms of the POC study, identify functional features enriched in these metagenome assembled genomes, or MAGs, and then to determine which bacterial pathways are in these WLZ correlated MAGs are expressed when children are treated with MDCF2 versus RUSF, and to correlate these gene expression data with results of analyses of the composition of MDCF2 and the metabolism by the microbiota by MDCF2. So identifying the bacterial WLZ correlated strains that are primary consumers of MDCF2 are important tasks ahead. those types of questions, those types of approaches, the search for bags, the use of microbial RNA-seq to go beyond just the DNA-based description of the response of the microbiota to a food-based intervention, also opened the door for a next level of MDCF2 discovery, returning to the strategy I described. Anna Lynn, an MD PhD student in the lab, is performing these sorts of analyses where different foods, complementary foods, consumed by populations in different parts of the world are screened for their effects on microbial communities from undernourished populations living in these parts of the world, where these food ingredients are culturally acceptable, have satisfactory organoleptic properties, and come from sustainable sources. An additional aspect to this flow diagram is a reverse translation experiment where microbial communities captured prior to, during, or after one of these POC trials can be re-enrolled in the trial in the form of their transplantation to notabiotic animals. And the trial conditions applied to the animals so that we can take a much deeper, more refined view of the signaling pathways that connect food metabolism, and the response of different cell lineages in different organs of the animal. So let me conclude. What are the implications? I hope that I presented some information to you that might persuade you to believe that healthy growth might be linked to healthy development of the gut microbiota, and that you have a more encompassing conceptualization of human developmental biology, one that includes a consideration of the organisms in gut community, the microbiota, and their vast repertoire of genes, the microbiome. A possibility for a better definition of wellness for infants and children that has implications for preventative medicine. We have at least a 2-year window in postnatal development to intervene if there's a alteration in the developmental program of this microbial community, and such an intervention may have very long-lived effects beyond the 2-year period. The implications also about how to feed our infants and children, what kind of complementary foods, informed by knowledge of gut community development, so that we can be much more effective stewards of our children's precious microbial resources. And finally, the need to determine the effects of interventions at different ages, earlier, the duration of the intervention, the durability of the effects of the intervention once it is stopped. So generalizability, durability, and safety. We're altering human developmental biology by trying to promote catch-up growth of the microbiota. Is that safe? Is that as effective? And this is something that we are pursuing at several different sites around the world where the burden of undernutrition is great. So I thank you for your attention.
Thank you very much, Jeff, for this really very exciting presentation and illustrating very powerfully how the problems of finding cause and effect between a clinical phenomena, in this case malnutrition, and the microbiome is a very difficult, challenging, and problem, and is really where the action's at. And it's very, I think, amazing to read of your studies and hear of them and realize that it's much more complicated than just presenting a pattern of distribution of the microbiome in a given population group or a given disease group and comparing it. The mechanics, the mechanisms are challenging. So we'll hold off on questions, and now we'll turn to Tim Greten. And Tim, would you please proceed?
Yes, sure. Just give me 1 second. Need to find my presentation that I can share. Okay, so I hope you can see my presentation now.
Yes.
Yes, sir.
So this is going to be very, very difficult after this presentation, but I will try my best. And we're changing gears quite significantly. We're going to talk about how the gut microbiome controls growth of liver tumors. Let me just put here my, oops, I was hoping it doesn't matter. So these are just a disclosure that I'm just going to, I want to show you some clinical data in the beginning, you know, how we actually got somehow involved in this. So, you know, as a general introduction, you heard already this, you know, the question really is, you know, how does the human cells in the human genome actually interact with the microbial cells and the genes that we have in the in every single body. And what we are now specifically studying here is the microbiome in human cancer. And there are certain aspects we know that we can use this actually for diagnosis. We can identify DNA in blood. We know that we can use this as a prognostic marker. There are different ways how we can actually utilize microbiota therapeutically. These are all ongoing research studies and how we can potentially use the microbiome to understand metastasis. Now, what I'm going to talk about is not cancer in general, but I would like to focus specifically on liver tumors. And liver tumors can be divided into either the majority of tumors that occur in the liver are liver metastases. And then there's also primary liver cancers that occur in the liver. The number one is the hepatocellular carcinoma, and the less common one is cholangiocarcinoma. Now, what I'm going to show you in the second part of my talk is mainly research studies that really look at the tumor microenvironment. It's going to be in the context of these two different diseases. But to be honest, it doesn't really matter what type of tumor we look at. The mechanisms that I'm going to explain to you are really something that we see primarily in this liver-specific environment, and they apply to everything, to every type of tumor. Now, why is there this interest in the gut microbiome and in the liver? And that's very simple. It's just the anatomy. You know that the majority of the blood that goes into the liver and feeds the liver with blood supply is actually drained from the large and the small intestine, as you can see here, that drains into the portal vein and then going into the liver. So basically, everything that is in the gut can potentially migrate here and be drained through the different branches into the liver. Now, let me just give you, because I want to tell you a little bit about a small case about biliary cancer. This is a rare cancer. About 8,000 people in the United States are diagnosed. There are very limited treatment options. It's a very dismal disease. And we did a study, and I'm not going to go into the details. I just want to show you, you know, what drives our research, where we used immune-mediated treatment for patients with different types of cancer, liver cancer, including biliary cancer. We treated them with immune checkpoint inhibitors, which activate the immune response. And the only thing I really want to show you on the study is that you can see that the treatment was really not very effective. You see, the majority of patients were only treated for a short period of time. Tumors grew. But then there is this one outlier. You can see this patient showed a clear reduction in tumor, and it's actually very long lasting. So if we look at this particular case, you can see here now the clinical course of this patient. So we started the treatment in this patient in 2016, and I can tell you that we have seen actually this patient a couple of weeks in the clinic, and she is still tumor-free, and she was treated with the same treatment than the rest population that we have in our group in this trial. But somehow something must have been very different. The patient developed an abscess in between. We treated the patient with antibiotics. Patient developed a colitis. And to be honest, we performed a lot of correlative studies. We looked at we performed obviously genetic analysis of the tumor, and we were never really sure why this patient responded and is such an outlier, and I still don't know this. So what I want to tell you is immunotherapy may actually work in primary liver cancers. I didn't show you any data, but it's an established treatment for liver cancer. But there are so many unanswered questions, and our interest here is really to study the immune microenvironment of the liver, and more specifically, how the gut microbiome can change this, and how it does this, and how it does this in the context, or in how it affects immune responses. So how do we do this? Well, we basically took use of a very simple model, MYC transgenic HCC model, where we can use MYC tumors in the livers of these mice, and they develop tumors over a period of time. And then a number of years ago, this was one of the first studies, we treated these mice with antibiotic that we added to their drinking water. And you can see here, these mice developed way fewer tumors. Now, I'm a medical oncologist. I treat a lot of patients with cancer. This was very hard for me to believe that just antibiotic treatment would actually reduce tumor nodules that you can see here. We repeated this experiment. We used other mouse models where we have tumors in the liver. And I can tell you this, the result was always the same. So we did what we usually do when we go into immunotherapy studies. We performed immune monitoring. So we looked at different immune cells in the liver. You can see the list down here: CD4 cells, CD8 cells, B cells, NK cells, etc. But there is one particular cell population where we noticed that it is an increase in those mice in red that were treated with the antibiotics. And we observed this accumulation of so-called NKT cells. So what are NKT cells? NKT cells share the properties of T cells and natural killer cells. They recognize a CD1D molecule, which doesn't present a peptide like we usually know from MHC molecule, but it actually recognizes foreign lipids and glycolipids. It's rather abundant in the mouse liver. It expresses CXCR6, and we can actually identify this T cell receptor by its V alpha chain. So we looked in our mouse model in more detail, and basically this is a flow cytometry analysis that we performed. You can see that the majority of these cells are CXCR6 positive. We can identify them. And yes, indeed, if we look in the liver of these mice, we actually find more CXCR6 positive cells. Now, again, I'm not going to go into all the details how we did this, but the important thing is to really look and remember the liver anatomy that I just told you, with the portal vein draining the blood from the gut. But where does the blood go then? Well, once it goes into the liver, if you remember the liver anatomy, it goes into the from the portal vein through the parenchyma into the central vein. And this is basically, again, a snapshot or magnification of this from the portal vein into the sinus. This blood has to travel through the liver. And the first border that it has to go through are so-called liver sinusoid endothelial cells. We looked at these liver sinusoid endothelial cells in more detail because we asked, how can the gut microbiome actually affect NKT cells? Now, I told you that NKT cells express CXCR6. So it was very straightforward to look at the ligand for CXCR16, which is for CXCR6, which is CXCL16. And it turns out that indeed, liver sinusoid endothelial cells from mice treated with antibiotics, and remember, the mice treated with antibiotics were the mice that had more NKT cells and fewer tumors, indeed expressed more CXCL16 than the water controls. We confirmed this by immunohistochemistry. So what I've shown you so far is basically using antibiotics to completely deplete the gut microbiome. In this case, we used different antibiotics so that we could reduce the microbiome by more than 99%. Caused an accumulation of CXCL16 message and protein in liver sinusoid endothelial cells, which then led to an accumulation of these liver NKT cells, and then blockade of tumor. I didn't show you all the details. Obviously, there were more studies in between here where we knocked out NKT cells or the CXCL16. But the question really is, and remains, how can the gut microbiome control chemokine expression in the liver, more specifically in the liver sinusoid endothelial cells? Well, it turned out that the bile acid metabolism plays a significant role here. So one of the important functions of the liver is to produce bile salts, which are basically released through the common bile duct into the small intestine and are responsible in part for digestion. And the important thing to remember is that 95% of the bile salts are actually reabsorbed by the small intestine and go back through the portal vein into the liver. Now, the second part is that the bile salts are actually metabolized by commensal bacteria. Conjugated bile acids are released. A bile salt hydrolase metabolizes the bile acids into primary bile acids. And then the primary bile acids are metabolized into secondary bile acids by a specific 7-alpha dehydroxylation step. And this is done by commensal bacteria. And there is actually a specific bacteria, a specific Clostridium species that is responsible for this step of metabolizing primary bile acids into secondary bile acids. So based on this knowledge, we looked at this mechanism in more detail. So we looked at the primary bile acids in the liver and the secondary bile acids in mice treated with antibiotics, and you can see that indeed there is an increase of primary bile acids and a reduction of secondary bile acids. So we used bile acids and treated mice with bile acids and asked, can we actually use bile acids to control tumor growth? So you can see the result of this experiment. This is our control. Mouse treated with antibiotic has fewer tumors. And then if we bring back the secondary bile acids here, you can see that we can basically rescue this, or tumors will basically develop, mice will develop more tumors again. And here you see the cumulative data. Now this is interesting, but this only tells you that bile acids can control tumor growth. What we're really interested in is the bacteria. So there were a number of studies, and again, I'm just going to show you a few of them where we actually looked now at the specific bacteria in these mice. And I told you that there is a Clostridium species, Clostridium scindens, that was known to be able to metabolize bile acids. So we set up a model where we can now treat mice first with antibiotics and then basically bring back the Clostridium scindens into these mice, the commensal, and look at NKT cells. And you can see that Clostridium scindens basically starts to metabolize bile acids again. And with this increase in secondary bile acids, we also find fewer NKT cells in the liver. Now, this is again interesting. We can now show that these commensal bacteria can control the number of NKT cells in the liver. But what it does not address yet is if bacteria can actually control tumor growth. And that is now shown here. You already know the model. So we have mice, we treat them with antibiotics. We challenge them with a tumor that grows in the liver. And then we bring back the Clostridium scindens into these mice and look for tumor growth. And you can see antibiotic treatment leads to fewer tumors. But if we actually infect these mice with Clostridium scindens, you can see higher numbers of metastases or tumors in these livers. And what I don't show you, obviously, this is in parallel leads also to a change in the NKT cells number in those mice, which goes down again, as well as changes in the bile acid metabolism. So I think here, this is a classical and one of the first examples where we now can actually pinpoint, at least in a model system in mice, changes to a specific immune function by commensal bacteria. So we know that under normal conditions, bile acids are released, and then we have the microbiota in the fecal pool. And the question then is, what type of microbiota we have? Are they able to metabolize primary bile acids into secondary bile acids or not? And this balance of primary to secondary bile acid actually can control the release of a chemokine by liver sinusoid endothelial cells in the liver. Again, these bile acids go through the portal vein into the liver, inducing expression of CXCL16, which leads to the accumulation of CXCR6 positive NKT cells, and these NKT cells can control tumor growth. Now, this is an ideal mouse model where we can study this, and the big question obviously is now, does this apply to human and patients with liver tumors? We don't know yet. We're going to study this. What I can tell you is that we know from large patient cohorts that there appears to be a correlation between the bile acid concentration, primary to secondary bile acid ratio, and the expression of the chemokine CXCL16. We have shown this. And a study from colleagues in Europe have actually shown that the same antibiotics that we used in our mouse model can actually also change the bile acid composition in peripheral blood from patients that were enrolled in a small clinical trial. So there's certainly some data suggesting that this mechanism may also apply to human. So we're testing this currently in a clinical trial where we basically treat now patients with vancomycin to target this specific Clostridium species, and hopefully in a year or so I will be able to show you this data. Now I would like to change gears again and go and tell you about biliary cancer. I already mentioned this at the beginning, to explain to you a different and a second mechanism how gut commensal bacteria can control anti-tumor immunity. Now, I told you that biliary cancer is a relatively rare primary cancer of the liver, and it is somewhat interesting for us because there are certain risk factors, including a disease which is called primary sclerosing cholangitis and inflammatory bowel disease, which are associated with biliary cancer. Now, it had already been shown that there is what we call intestinal dysbiosis in patients with primary sclerosing cholangitis. You can see this here. You see on this PCA analysis in green the healthy controls, and then here patients with PSC or patients with PSC and inflammatory bowel disease. And here you can see the more detailed description of the commensal bacteria in these individuals. The important thing to remember here is that apparently a disease in the liver can cause dysbiosis and changes in the composition of the gut bacteria. So we decided again to bring this into mouse models where we can study this in more detail. We use two different cholangiocarcinoma models, one that is dependent on AKT and Notch, and the second one that is dependent on AKT and YAP. And the way we do this and the way we use this is by a technique which is called hydrodynamic tail vein injection. So we can actually inject plasmids of interest into the tail vein of these mice. And because we use a relatively large amount of volume that we inject in a short period of time, we can get a significant and high amount of transduction efficiency in the liver. And these mice will express the gene of interest, and in this case, it's the oncogenes, and they will develop cholangiocarcinoma. We combine these models with different PSC models, which one is a genetic model, an MDR2 knockout model. You can see here how these mice develop these inflammation and scars around their bile ducts. And another model here on the right, which is a model where we perform bile duct ligation in these mice. So if we induce PSC in these mice, and you can see here the result from the mouse model where we induce the PSC by bile duct ligation, and the same applies to mice with MDR2 knockout mice. It was actually interesting to see, if we look at the cecum of these mice, that there are clear signs of colitis. I think it's very obvious to see that this picture here differs very much. You don't have the nice structure and epithelium anymore here in the mice after bile duct ligation. And if you look at tight junction proteins, in this case occludin, you can see that they are actually reduced in mice that have undergone bile duct ligation. We looked at a number of cytokines. Here I'm showing you the data for IL-17, which are increased in the gut of mice. We looked at the portal vein of these mice, and here we can actually identify 16S RNA back as a readout for bacteria. We performed studies with fitsidextran, which is a functional experiment to show you a leaky gut. So fitsidextran is basically fed to these mice, and then we can find this in the periphery, and you can see an increased uptake in mice after bile duct ligation as a readout for leaky gut and colitis. And if we look at patient samples, we can actually also find signs of bacteria and LPS. Now, if we look at the immune cell composition of these mice in which we induce PSC, we notice that there is an accumulation of so-called myeloid-derived suppressor cells. So these are myeloid cells which we had studied, or which we've been studying for a number of years, and which we know are very important immune suppressors. You can see an increase of this cell in mice with PSC. We can further subdivide, and I'm not going to go into the details, these MDSCs in so-called monocytic and polymorphonuclear MDSCs, and the majority of cells that we find are polymorphonuclear MDSCs. This does not only apply to one model, but we can also find this in the second model. Now, if we treat these mice specifically with neomycin, you can see that the accumulation of these myeloid-derived suppressor cells is no longer present. And if we perform FMT studies, so these are studies where we take regular mice kept under SPF conditions, and we treat them so that we either with vancomycin to target gram-positive or neomycin to target gram-negative bacteria, harvest stool samples from these mice, and then transfer these into germ-free mice. You can see that this actually has a significant impact on the number of these myeloid-derived suppressor cells. And you can see on the lower right corner that if we treat these mice with stool from vancomycin-treated mice, there's a significant increase, and we don't see this with the stool samples derived from neomycin-treated mice. Does this also lead to an accumulation of MDSCs, as shown on this slide? Now, the whole mechanism appears of accumulation of hepatic polymorphonuclear MDSCs appears to be dependent on, again, a mechanism of TLR4 and CXCL1. I can show you that these MDSCs are CXCR61 positive. Mice treated that have either PSC or treated to induce colitis have increased amount of CXCL1. We can block CXCL1 or overexpress it to control the number of polymorphonuclear MDSCs. And this is dependent on expression of TLR4 and hepatocytes. You can see down here in mice where the TLR4 receptor is knocked out, in hepatocytes, we don't find these cells. So what I've shown you so far is that in the context of liver cirrhosis, mice develop a leaky gut. We believe that LPS plays a significant role that comes into the hepatocyte, or hepatocytes start to produce CXCL1, which leads to an accumulation of, in this case, CXCR2 positive polymorphonuclear MDSCs. And this mechanism appears to be dependent on gram-negative bacteria. Remember the data that I've shown you with vancomycin versus neomycin. It depends on the TLR4, and that's why it's highly likely that this is LPS dependent. Now, I haven't shown you really anything about tumors at this point. So this is all only liver cirrhosis, leaky gut, and polymorphonuclear MDSCs. So what I would like to show you now is if we combine the models where we induce tumors by injection of oncogenes, in this case, in combination with hydrodynamic injection, we indeed see that these mice develop more and larger tumors. The same applies to a second model where we use now MDR2 and oncogenes, or if we induce colitis. But basically anything that I've shown you in, so far that controls the MDSCs also controls now tumors. So if we block MDSCs, we find fewer cells. If we overexpress CXCL1 in the livers, we find more tumors. If we block the MDSCs using an antibody against CXCL1. If we perform similar FMT studies that I have just shown you, we can actually control again tumor growth in these mice, dependent on whether we colonize mice with stool derived from mice, initial mice treated with vancomycin or neomycin. And this entire mechanism, again, is dependent on expression of the TLR4 receptor on the hepatocytes. You see if the commensal bacteria or the LPS derived from these commensal bacteria in mice with colitis go into the liver, and the hepatocytes do not express the TLR4 receptor. We have fewer tumors, and we can basically overcome this again by overexpression of CXCL1. And this mechanism of tumor cell growth inhibition appears to be dependent on NK cells. So this work that was primarily done by a postdoc in my lab, Shanfei Zhang, is basically summarized here. So under normal conditions, there is shown here a normal composition of the microbiota, but once through whatever mechanism you change the liver and induce fibrosis, this will basically change bile acids and cause other changes that will lead to dysbiosis and a change in the composition of the microbiota. Now, this change of the microbiota in the intestine will cause a leaky gut so that bacteria and bacterial products can transmigrate into the portal vein. And then what I've shown you here, that through a TLR4 dependent mechanism, hepatocytes express CXCL1. The CXCL1 consecutively leads to an accumulation of these CXCR2 positive granulocytic MDSCs. I told you these are cells that can basically cause or suppress immune responses and then thereby enhance tumor growth. So I believe I've shown you here two different models how the microbiome can contribute or change the tumor microenvironment and contribute to hepatocarcinogenesis. I've shown you how the leaky gut can lead to an accumulation of DAMs and MAMs, which can cause inflammation. But there's other mechanisms that I didn't show you out of time, and because I wanted to specifically focus on immune responses, we know that there are also effects which are mediated by stellate cells. We know that the bile acids which are affected in these, or which can be affected in this system, can also have direct effect on hepatocytes and mTOR mechanism, as well as other mechanisms through short-chain fatty acids exist, which have been described to control tumor growth. So what I would like to summarize and leave you with is, there are microbiota dependent, or there is a microbiota dependent regulation of cancer development, progression, and treatment. In this particular talk, I told you about the commensal microbiota in the gut and how they talk to the liver. We know that these microbiota can be changed through different mechanisms. You already heard in the first talk how diet and can change this. There are certain host genetics, lifestyle, etc., leading to dysbiosis that I have shown you. We can potentially change this by antibiotics. I've shown you this in our animal settings. There are xenobiotics. We can perform fecal transplantation, and this is what is done in clinic for other indications. This has effect on different immune cells in different parts of the body. In my talk, I specifically focused on the immune cells in the specific environment of the liver, and I showed you in the first part changes on NKT cells, and in the second part how it affects myeloid cells. And this then ultimately will lead to changes in cancer initiation and progression. But there are also very good and interesting data how the gut microbiome can actually potentially enhance the efficacy or impair the efficacy of different types of anti-cancer treatments, including immunotherapy. And there is accumulating data on comorbidities such as cachexia and potential side effects. And with this, I would like to close and thank the people in my lab. The first study on the NKT cells was primarily led by Chi Ma in the group, the second one by Shan Fei Zhang. And none of this work would be possible without collaborators within the NIH, Giorgio and Tiankeri, Xin Wang, and Jay Berzovsky, who provided important help, and outside collaborators that provided reagents, samples, experience, etc. And with this, I would like to close, and I'm happy to take any questions.
Well, thank you. Thank you very much, Tim. So now we have a whole series of questions, and I've tried to put some of them together so that folks, not everyone who has submitted a question, I think, will actually be able to answer or to present. But let me pose a few of them here. So, Jeff, the intestinal microbiome consists of more than just bacteria. And there's an interesting question about the possible role, or what do we know anything about the role of phage as presumably the major virus present in the type of virus present in the intestinal microbiome. Is phage likely to be the next area of concern in terms of its influencing microbiome activity? Or would you comment about that? It's an interesting question, I guess.
Well, I think that that's a very appropriate question to ask, considering that phage influences the composition of bacterial communities, that many bacteria have prophage introduced and maintained within their genomes. The parts of the microbiota are interconnected. It's a very dynamic system. There is, in a healthy microbiota, robustness, the ability to express certain critical functions despite a number of perturbations, and resiliency, the ability to recover from perturbations. There's little question that the phage dynamic is constantly playing itself out within the gut microbiota, but it's an extremely intricate and difficult system to be able to dissect. There is an effort to characterize the phage that are represented in microbial communities of different individuals representing different ages and health states. Many of the genes in bacteriophage encode proteins for which we have little understanding of their function, which limits, of course, our ability to greatly appreciate their influence on their host bacterial cells. Phage have been viewed for quite some time as potentially a therapeutic agent, so that one could deliberately stage a phage attack and perhaps perturb the microbiota in ways that might be healthy or target particular organisms. I would ask the person who posed the question to consider the fact that phage are a component of the diversity present in the microbiota, are instrumental in propelling the dynamism of the microbiota, but we have an awful lot to learn. I would say that one school for learning lessons could be the very notobotic animal models that we talked about, where defined communities of microbes can be introduced into the intestines of these animals, and so-called defined communities created, and deliberate attacks on those communities by phage, recovered from healthy or diseased individuals, can be used to start understanding not only the immediate perturbations produced by phage, what are the tropisms of the phage, which organisms do they connect to, but the development of phage resistance. This is a form of bacterial immunity, whether the phage dynamic is more temperate or not, and what the factors are that affect whether prophage go lytic or not. So there are lots of questions evoked by the very question of the person who listened to this lecture.
Another interesting question is, in some of your earliest work, I seem to recall, you were interested in the question of how the intestine actually develops, how the individual cells carrying out different functions along the length of the intestine sort of compartmentalize those areas of the gut. And so there's a question as to what determines, well, it's a double question. What determines the microbiome development with age? And the other side of it is, does the microbiome in the newborn play a role in this development of the intestine and its various segmental functions and distributions?
Biogeography, the fact that we have to look at the intestine as changing not only with age during the first several years of life, but also the fact that there are different community configurations along the length of the gut. That the small intestinal microbiota has been a terra incognita because of the difficulty in sampling it. That when we think about repairing a microbiota that's been perturbed, we have to think about the age of the host. We talked about the co-development of the microbiota and the host, and what regions of the intestine we want to seek to repair. I'll just say to the person who asked the question a couple of things. Number one, there's an ongoing effort to determine the degree to which the small intestinal microbiota is contributing to the pathogenesis of undernutrition. There's an enigmatic disorder called environmental enteric dysfunction that was first identified in Peace Corps workers returning to the United States after living for several years in areas of poor sanitation, where they had developed malabsorption, weight loss, diarrhea, and they were endoscoped. And what was seen was that the small intestinal villi had collapsed. The surface area of the epithelium had therefore been reduced. The epithelial barrier had been disrupted. There was a chronic immunoinflammatory disorder in the small intestine. The Bill and Melinda Gates Foundation has been wondering, as many people have been, whether the small intestinal microbiota is perturbed in children with undernutrition. Recent studies have been performed of children who failed standard interventions, and where in highly specialized centers with ethical approval, these children underwent endoscopy to determine whether their small intestine had the perturbations that were seen in the adult Peace Corps workers. And yes, in fact, many of them did. The small intestinal microbiota from those individuals, when reintroduced into germ-free animals, can reproduce this enteropathy. And I would say that there is no question about the fact that as we move forward in this field to look at the developmental biology of the microbiota and repair in states of disease, that we're going to have to be able to have a positional perspective about this. Now, one last comment. And the person who questioned, who raised this question, must have spent some time in the lab under stealth technology, because this is a question that we're asking and addressing right now. So is the repair of the microbiota that we're seeing in these children associated with effects on, for instance, epithelial biology? So can one connect the dots to see how repair of the microbiota reads out in terms of the expression of genes in different epithelial lineages as they execute their differentiation program along the crypt-villus axis, or if you're in the colon, along the crypt to surface cuff axis. And there's a lot of reason to believe that that is one manifestation of the impact of the microbiota, just like we believe that the microbiota shapes in healthy individuals the development of the epithelium. Everything is connected. There's co-development is the watchword of the microbiota, with many aspects of host cellular and organismal biology.
It's been asked whether there's a correlation between the gut microbiome and the skin microbiome in terms of changes, perhaps during development, I guess is what the question was being asked here in the newborn. And if so, can studies of the skin microbiome provide clues as to what's going on in the intestine, and maybe make things easier in terms of assessing some of the clinical studies?
Well, I think that the statement is a very good one. There is maternal transfer of microbes to a neonate. Some of that transfer involves organisms associated with skin. The skin is a very biogeographically complex organ with many different and varied metabolic features, depending upon where in the skin we're talking about. And that's reflected by differences in community composition and function. The often cited example of the role of the skin in maternal transfer of microbes to the infant is the differences in initial gut community configuration in vaginally delivered infants versus those that are delivered by cesarean section. I think that the precise role of the skin microbiota in defining the later trajectory of development of the microbiota in the gut is not as well understood. But to the extent that there's co-development of microbial communities in the immune system, and since the immune system is also important as a factor, as a determinant of community configuration, one could imagine that there are very many ways in which these two communities might be connected.
several people asked, what are the plans for following up the participants in the Bangladesh study in terms of following them through many years to find out whether there are things in their cognitive ability or any other measurable event? Is that part of your plan?
Well, I love the question for many reasons, because it emphasizes the importance of longitudinal studies and understanding how the microbiota affects host biology, rather than cross-sectional studies, how much more informative longitudinal studies are. I love the question because it speaks to the point of what are the effects of repair of a microbial community on host systems biology, and how long is the repair evident in terms of the configuration of the microbiota, both in terms of cellular and molecular configuration, metabolic configuration, and whether the effects on different organ systems also are durable. So we feel that it is critical to do these sorts of follow-up studies in populations where the level of trust and devotion between the healthcare providers and the families is enormous. And that is one of the reasons that we're so fortunate to be able to interact with our colleagues at the ICTDRB. And yes, the proof of concept study of children with primary MAM who lived in Mirpur is being extended for 4 years with every 6-month sampling. Even though it's only a 3-month intervention, we feel that those sorts of analyses of both the plasma proteome and also of the microbiota are critical. We would love to look at much more detailed phenotyping, including, as the person asked the question implied, neurodevelopment, long-term effects on body composition, on immune responses. Those types of studies are very difficult, as most clinical investigators can appreciate, to actually execute, and are quite costly. We feel, on the other hand, they're critical, and they also have to be sufficiently powered. I'll say one other thing, not to be too loquacious, and that is the importance of determining whether a more extended period of intervention is necessary to have a more durable effect, and what the effects of interventions are at different points in postnatal development. Would an intervention for 3 months in a child who is 6 months old have a greater impact, greater effect size than later on? On the other hand, you can say the microbiota has many different functional states, and there are many different biological states to being undernourished. And I think that one of the important lessons that we want to learn in this area is how repair of the microbiota is, we will define this repair much more cogently, and to look at how different initial states of the microbiota may have greater effects on certain cellular and organ systems than others, depending upon the starting state, biological state of the child, including their age as one factor.
Your studies are in malnourished, very young children. Do you think that a similar mechanism may be afoot in much older individuals who have diseases which are characterized by weight loss and maybe even maintaining a diet, but they're still losing weight? I'm thinking of patients with liver disease in particular, but to try and bring it in to perhaps Tim would want to comment. Is it possible that what you have observed, Jeff, in young children is also occurring in much older individuals, perhaps even in aging, let alone with concomitant major organ disease? What do you think of that?
you want Tim to answer, or do you want me to answer? Whoever.
Maybe you can both answer if there is an answer. Tim, what is your view?
Well, so I mean, obviously, you know, that there may be similarities. I'm sure there are similarities. You know, it's very difficult to, you know, it's very difficult to predict this without doing the study. But I have a question actually, and when Jeff is going to give his answer, you know, which is in the same direction that was just asked about the long-term follow-up. We know that any perturbation of the microbiome, so anything we do, is very rapid, but usually not long-lasting. And I saw on your slides that you actually follow the children for, I believe, I don't remember if it was 1 or 3 months after you stopped the intervention. So do you think, you know, you're going to have long-lasting effects in those children?
One of the reasons that we wanted to begin these types of studies in the developing phase of the microbiome when it's first formed is that if you believe in the hypothesis that co-development of the microbiota and the host has a profound effect on setting physiologic state or physiological trajectory, it's entirely possible that as these homeostatic mechanisms that exist are first established to control cellular biology, organismal biology, that you could have a durable effect. So I do think that it'll be interesting to see the duration of an effect of microbiota repair during its formation, as opposed to the effects of repair once the organ system and the host has fully matured and developed. So what is the effect of a repair to the microbiota in a 60-year-old as opposed to a 6-month-old? So,
if that is the case, the answer to the question, if you will see the same effect in an adult that you see in these small children, is probably no.
Maybe yes or maybe no. If one goes beyond the types of studies I've just described to a deep and inspiring, but nonetheless difficult task of identifying signaling pathways by which different components of the microbiota influence host biology. So if you're able, as a durable legacy of these types of experiments, to ultimately, or studies, to ultimately be able to identify these signaling pathways, those pathways and their components become your therapeutic targets. And there is reason perhaps to conjecture or to postulate that those pathways still operate during the course of an individual's lifespan. So I think that these are, I think, one of the agenda items. If we're fortunate enough to be able to be supported in these types of translational studies and reverse translational studies, to be able to discern the signaling pathways and their components. That's my hope and expectation. But I also agree with you, sobriety is very important in this field, and to test the generalizability of an intervention. Children, for instance, of a particular age living in different regions of the world who present with similar biological states, to be able to determine the durability of the effects, that's absolutely critical. And I hope that's done in a very cautious and methodical way.
Tim, there was an interesting question where someone has pointed out that the vast majority of patients with hepatocellular cancer have some underlying chronic liver disease associated with a lot of regeneration, cirrhosis, all sorts of things. And the question is, does dysbiosis in the intestine, is that considered to be a major route leading to fibrosis, altered hepatocellular regeneration, and not directly to cancer itself. How does one, can you visualize a way in which one can differentiate these various associated events with the fact that the cancer is occurring in patients with chronic active forms of liver disease, fibrosis, and a lot else going on?
Yeah, I mean, obviously, that's a very interesting question, and obviously this will be impossible to do in a patient, but there are experimental models where you can do this because we have ways to induce dysbiosis. And I've shown you our mice with the liver disease that have significant dysbiosis, but there's many other ways how you can induce dysbiosis. Just simple antibiotic treatment, for instance, is one. And then if you go into the animal system where that allows you to do FMT studies, you can actually induce the FMT in germ-free mice and then look at the effect on mechanisms that will ultimately lead to fibrosis and cirrhosis in mice. So there are certainly ways to do this. Now, unfortunately, I have to say it is not as straightforward as one would think, because germ-free mice actually have significant changes in their GI tract by itself. So just the absence of bacteria is actually already causing significant changes. But yes, we do know that dysbiosis can have significant effects. I showed you some of them. So it is believed that can either induce or amplify changes in the liver. And this is not only in the context of viral infection. We know that coming back to the idea of nutrition, that obviously not only with malnutrition, but also with obesity and fatty liver disease, there are significant changes. And we know that the gut microbiome in those individuals is significantly different. So the idea that the gut microbiome plays a significant role here is clear. And then last but not least, there is very good data that the gut microbiome can actually also control alcoholic liver disease. This is done work primarily from my collaborator Bernd Schnabel at UCSD, who has done this work. And the reason why I'm mentioning this because you talked earlier about the phages, and he has actually shown that you can use phages to potentially also disrupt mechanisms thereby and thereby treat alcoholic liver disease. So it's a little bit of a long answer, but basically, they are all interconnected with each other, and it's very difficult to say in the end, which is the hen and which is the egg.
Do we understand how any intestinal microbe causes dysbiosis? Is that understood? The mechanism.
Well, I mean, I'm probably not the right person to answer this, but there's a competition between different bacteria. So if you kill one, another one will grow out. That would be my very simplistic answer.
What do you think?
Wynn, I think that one of the lessons of this area is how elaborate the interactions of microbes are, and that microbes don't function in isolation. And I think there's been a lot of work, speaking of bile acid metabolism with C. difficile, and how very complex metabolic interactions occur that are responsible for its ability to move from a pathobiont to a pathogen and cause severe pathology. So I think that we can think about microbial communities as health promoting or disease promoting, depending upon the biological context in which they exist. So I think we have to have a much more transcendent view of things. Just like the nutritional value of food may be influenced to a significant degree by the chemical composition of food. So the microbes don't see food as broccoli. They see it as a complex milieu of glycan structures, and different organisms are capable of having different roles in the processing of these things. Although in general, these communities have a lot of strain level diversity and redundancy that allows them to withstand different perturbations. This evolved as an adaptive metabolic organ. The number of genes in the microbiome vastly exceed the number of genes in our human genome because they are able to provide us with biochemical capabilities that we just have not had to evolve on our own. And I think that that's one of the ways that we can construe this symbiosis, bringing together of organisms that have different jobs so that the holobiont, the entire organism, is fitter. That's the miracle. How do these systems assemble? How are they maintained as we perturb them? How do they recover? It's stunning and inspiring, but trying to figure out the underlying mechanisms really requires a lot of tools. And luckily, we live in an era where we have the capacity to do deep sequencing, not only of DNA, but RNA, and do a lot of metabolomics. It's and have model systems where we can reduce complexity to start learning basic principles that glue organisms together and to connect them with ourselves. So that's the hope, but humility, given the face of this complexity, and the lack of hubris and sort of sobriety is really important.
Thank you. Tim, you mentioned a moment ago that in germ-free animals, the intestine is not like it is in infected or normal. You want to comment briefly about that? What are the differences?
Any postdoc or graduate student in the lab who is working with this only has to open the abdominal cavity of a mouse, and you will immediately see gross changes, significant changes. You will see a hugely enlarged cecum. You will see the gallbladder differently. So there is just, you can see this with your bare eye. Obviously, once you start looking closer and doing microscopic, genetic, etc. analysis, you will see, more changes. But it's very, very, very obvious, and it's stunning when you see it the first time.
It's very exciting. Well, listen, we're out of time, and we want to. I'm sure, on behalf of the many, many, many people around the world who've been taking part in this, they would join me in thanking both of you for taking the time, the interest, and presenting such really provocative and challenging, exciting opportunities to build bridges. And it seems as if there are great opportunities in these fields for bright young people who are looking for new worlds to conquer. So thank you very, very much, and good luck.
Thank you.
Thank you very much.